and welcome to Trait Talking. I'm Dr. Stuart Grice. Today we are going to talk about epigenetics. But let's start with a post I've seen, which I think underlies a lot of the misconceptions about epigenetics. So let's have a quick read through. Obesity is not about genetics, it's about epigenetics. The foods you eat and your environment will be the main tools in how much fat you gain or lose. So let's just pick this apart for a moment. And I think the three issues I have with this. One is the uncoupling of genetics and epigenetics. That's saying that they seem different processes. You can have one without the other. The second is the placing of epigenetics above genetics in the hierarchy of the processes in the cell, which then confer risk to obesity. And then the third is actually the sleight of hand where you're saying epigenetics is the environment and the environment is epigenetics, and that's just not the case. So I'm going to sort of use this as a tool to describe and go through what epigenetics is and what it means in terms of the context of the cell and what it means in the context of, say, a condition like obesity. But first, let's kind of, a bit of housekeeping. What is epigenetics? There are a number of definitions that vary around epigenetics, but I'm not really going to pick any fights with those, but we just need to be on the same page. When I'm talking about epigenetics here, it's the study of how organisms or cells control gene expression without changing DNA sequence. So we're not talking mutation here. It's some way that the cell can control or change gene expression without modifying any of the bases in the DNA. So what we're going to do, so we're going to just talk about gene expression in general, quick 101. Then what is epigenetics in a broad sense? I'm not going to go into a lot of the sort of chemistry, but just what it means in terms of gene expression. And then we're going to address the point what was in that post. And we're going to go to, we'll really break down what genetics and epigenetics means in context with each other. So DNA expression, gene expression. So obviously our DNA has many genes in it, and these genes are expressed in terms of protein expression through a molecule called messenger RNA, but also a lot of genes actually express functional RNA. So these are RNAs that may have catalytic properties that help with gene expression or the processing of messenger RNAs that then go on to form proteins. So this is kind of known as the central dogma, and it's been known now for over half a century. There are many proteins and also many functional RNAs that are encoded by the genome, and we really need to know ways of how do we switch these on and off, when do we express them, and there is information within the DNA that can help us do this. So if you were to zoom out from your gene, so as you can see here on the bottom bit, you've got the gene, a representative gene. It seems quite small for a gene, but bear with us. There are regions of non-coding DNA that contain sequence motifs that can bind proteins. Now we've got promoters that are generally close to the gene, and these can bind the transcriptional machinery that will then create the messenger RNAs from the DNA sequence. And further away, and these can be many kilobases away, you get enhancers. Now these can bind specific transcription factors that actually can create tissue specific or time specific expressions. So kind of how does this look in terms of regulation? So just a little point here, where we know that proteins and functional RNAs actually can regulate the gene expression. So at this stage, we're not talking about epigenetics, but the components of the DNA that are coded by the DNA actually can drive proteins and RNAs that will then bind to the DNA itself or to the messenger RNAs to process it. So going back to our promoter and enhancer complex, here you can see DNA exists within three-dimensional space. We're so used to seeing it as one big long string when we represent it in PowerPoint presentations. And you can see here how, even though far away, the enhancer and the promoter are now in proximity, and the enhancer binds some proteins that then coupled with the transcriptional initiation complex that then sits it on the promoter, which now enables transcription of that gene. So at this moment in time, we are driving gene expression, and it is proteins that are driving that expression of the gene. Now, but we're here to kind of talk about epigenetics, aren't we? So other things that happen as opposed to proteins binding and transcribing the genes is you can get chemical modifications on the DNA, and this can happen in the regions we've seen in terms of promoters and enhancers, etc. Not going to go into the chemistry of this, but there's kind of 3 levels of epigenetic modification. 2 of them are modifications of the chromatin or the DNA itself. So we get molecules on the histones, these the proteins that DNA is bound to and wound round to create it compacted into chromatin. These can be modified either to open the DNA regions up so they're accessible to these transcription factors and transcription initiation complexes, and now enabling them to be transcribed, or they can be closed up so they're inaccessible and therefore not expressed. The second one is DNA methylation, which is probably the one you've most likely heard of, and this is where you get methyl groups attached to the DNA, and this generally represses gene expression. There are always caveats to the rule in biology, but generally represses gene expression. And the last one is non-coding RNAs. So again, these are encoded by the genome. So these are not chemical modifications. They would be transcribed from genes or long regions of non-coding RNA. And these can interact with the DNA itself, proteins associated with gene expression, or RNAs. So there are many ways that non-coding RNAs can actually affect gene expression, and we won't really go into that today. So it's quite a new field in terms of epigenetics, a very exciting one, but this is something we won't go into detail. And I'm going to use methylation as kind of the primary example through this to discuss what epigenetics is. So if you remember a few slides ago, we had our transcription complex and our enhancers sitting on the DNA, and our gene wasn't expressed. Now, what can happen is we can get epigenetic modifications, particularly around the promoters and enhancers, but it can be in other regions of the non-coding DNA, that stop these factors binding. So this is a way, even though you may have open chromatin, for these molecules to to stop the transcription initiation complex from binding and transcribing the gene. So in this case, the methyl groups are stopping this gene being expressed, and this is really one of the fundamentals of how these processes are stopping the transcription of a gene. But because it's a molecule attached to the DNA and there's no change in the DNA, it is not through any change in the bases. So here's the fundamental part of what epigenetics is. Now let's think about the diversity in our body, and I think one of the most interesting, exciting components of how epigenetics regulates gene expression is in terms of development. Now, if you think about our body, we've got many specialized organs and millions and billions of cells within these organs that have to express different sets of proteins to become specialized, different transcription factors that create the cell identities, different molecules that then can assign the function to the cells within those particular organs. And one of the great questions is, in developmental biology over the last 100 years, is how is the transcriptional and ultimately protein diversity created in these cells, these unique sets of proteins and messenger RNAs? Well, first, it is actually the proteins, the transcription factors, as we talked about, enhancer complexes. These combined tissue-specific transcription factors and enhancer complexes that are then going to create other proteins that will bind to genes specific to that tissue. So you may get a brain-specific or neuron-specific transcription factor that then binds to other genes that then transcribe neuron-specific genes, and you can think about this in many ways. But what you really want is a way of codifying this and actually creating a robust repression of some genes and expression of other genes, so we don't have to have this constant cycling of binding and an interaction with the DNA. And this is kind of where epigenetics is a great tool. So if you think about, here is a heart, we've got a heart muscle cell nucleus, and excuse the meiotic chromosome there, misrepresentation. But in this cell type, we've got 2 genes. You've got a heart muscle specific gene and a neuron specific gene. The neuron specific gene has methylation groups associated with the enhancers and the promoters, and this gene is not expressed. But the heart muscle doesn't, and this gene is expressed. So your producing heart muscle specific genes at the right time, in the right space, and the epigenetic regulation, the epigenetic markers can codify this, which enables the cells to be expressed in the right genes. So it is really a great endpoint, particularly when the cell is differentiated, to make sure we're driving the right genes to create or maintain the identity of that cell. But probably where you guys are more interested is, and again in the health space or the fitness space, there's this excitement around actually the transient nature of gene expression and how environment can lead to epigenetic changes. So this can be exercise, it can be infection if you smoke, all the different things. These things can directly impact your epigenetics. And again, this go back to the post at the beginning where it's all about epigenetics. It is your epigenetics that is causing you to be overweight, and it's the environment that is directly interacting with these epigenetic signals. So just a bit of housekeeping at the moment. So epigenetic modifications change gene expression. And we've got into a bit of detail here where we show, as the cells grow and differentiate into specific types, this is really important. This is a way of defining the lineage and the identity of that cell. And as you can imagine, if that goes wrong, that has significant implications early in development. The second one, which we're going to talk a little bit more about now and for the rest of the presentation, is the response to environmental changes, because this is really interesting in terms of gaining weight, disease, how we age. I think this is why this is quite an exciting field to go into. But there's a caveat, and I think this is really the point in terms of understanding the science. Is we've talked about, in this case, obesity not being genetics, about epigenetics. This requires a significant amount of, places burden on the statement that the epigenetics is the primary driver of these changes in the cell. Or is it in fact the secondary mediator of the adaptive response? As we've seen with the differentiation of the cells, you have the transcription factors arrive, they derive the identity of the cell, and at the end, the epigenetics then codifies that. But there's a big process that sits in front of it. So the burden of proof on the epigenetic statements of it is epigenetics, not genetics, needs to prove that environmental change or environmental insult, so lots of food, lots of fat, smoking, etc., toxins, can directly interact with the epigenetic marks on the chromatin or on the DNA. And So, and this would be the proof that epigenetics is the primary modulator of this gene expression, and therefore the statement, it is epigenetics, not genetics, can therefore be proved. And unfortunately, there's currently not a lot of documentation on any mechanism where the environment directly interacts or communicates with the epigenome. There's nothing that exists. And in reality, the signals from the environment excuse me, are augmented through many layers of regulatory proteins. So if you think about, you eat food, you get an insulin spike. This tells cells to take up food. The food's processed in the cells, and also there may be other signals that then, if this becomes a chronically overeating, this may then lead to the cell to make a decision to change the expression of a gene. Then proteins that interact with the methyl groups, or lay down the methyl groups, or drive the non-coding RNAs, or modify the histone acetylation or the simulation or ubiquitination. They then drive the epigenetic changes. Now, I think this is a fundamental point to be made, that it is the proteins and the pathways that are associated with these proteins in the cell that actually derive the response to the environment. And of course, these proteins are derived from the genome and are subject to any mutations that can also impact their regulation, structure, or function. So therefore, genetics is in essence one of the driving forces for the response to the environment. However, epigenetics can then codify and reinforce. So epigenetics and genetics are not independent. They act together. Genetics technically is one of the primary drivers of the variation in how we respond to things. However, epigenetics can then validate and reinforce that. But also as well, the environment is really key here. And the environment sits both, above, in a way, the genetics and the epigenetic regulation in the context of chronic lifestyle diseases, at least. And I think this is a really important point that we need to tease apart, the fact that the genetics is fundamental to this variation in response to environment. And you can kind of see this as well with when you start to look at conditions where epigenetics is a problem. You see, you look at the chromatin or the DNA, you can see switches in epigenetic markers, or some genes are overexpressed or underexpressed wrongly that leads to disease because of changes in epigenetics. And all these diseases are because of mutations in genes that are involved in the augmentation or the laying down of epigenetic signals. Now, of course, these are monogenic disorders, so they're sort of pronounced and maybe not representative, overly representative of the sort of more chronic diseases. But this shows, in essence, how genetic variation impacts the epigenetic outcome. And I think this is a very important point when talking about that uncoupling. So let's go to obesity now and think about it in the context of what makes us fat. So here is a little sort of schematic of genetic risk, where at the top you've got high genetic risk, at the bottom you've got low genetic risk. And I'm not going to go into the genes or pathways involved in predicting this. We've done a podcast on this, Why Losing Weight is So Hard. If you're interested in the genetics of obesity, please go to that. But what we're going to say here is the genetic risk is quite well defined. People with very high genetic risk have a very high probability of being overweight, but also, interestingly, people who have very few of the genes associated with increased risk are generally quite lean. So we have this nice distribution that shows that actually there's an association of risk on the spectrum that's well defined. And of course at the top you get people with monogenic forms, and then generally in the middle if you've got a strong burden, it doesn't necessarily mean you're going to be obese, but the risk massively increases. And I think this is a very important point when you start to place environment on there. And when we're talking about the environmental component, which the post placed on, it's a very important point that the environment is key here. So I've decided to use pre-1970s as our non-obesogenic environment. I'm not quite sure if that's the cutoff, but of course we had the build-up of processed foods and sort of different working methods in the 80s, really. So that's why I picked. Any social historians or historical epidemiologists can post and tell me I'm wrong, but I think generally the point here. We had a non-obesogenic environment, so we had lots of people with higher genetic risk, but generally it was only the people at the top who had very significant reactions to food, being their desire for food had been incredibly strong, because a lot of the genetic risk is behavioural. It changes the brain and the way you respond to the food, the way you feel full, the way you pursue food. But generally, most of the population was not overweight. And I do love, every so often, there'll be a video of 1950s London or 1950s New York or California, and you'll always have someone post. Obviously, there's some more nefarious posts you generally get, but there's always someone post, "Look, there's not a fat person there," and that's kind of true. There would have been fat people, but they've been in the minority. And it's because, even though genetic risk exists, the environment didn't exist. Now let's move to post-1970s obesogenic environment. Now what we have is a lot more overweight people. The environment has very much changed, but these people still display obesity risk on the spectrum of the genetic risk. So the higher the genetic risk, the more likely you are to fit within this obesogenic, the obese groups or the overweight groups. And again, with the people who are underweight, generally what you see is these people are not underweight, sorry, are lean and not overweight. These people are generally found in the lower risk categories. And this ultimately really shows this genetic environment interaction. It is not epigenetics. It is a genetic environment interaction, and a complex one, where you have this burden of genes that is making you more likely to overeat. Non-obesogenic environment, it just doesn't materialize. You don't get overweight people, but you place people in that obesogenic environment, then the genetics is going to drive them to eat more than their peers with the low risk, and you then still get this distribution, but it's key to the environment. So back to the post, it is genetics. But also they're right. The environment is the key component. But understanding the genetic lesions and the genetic mutations and how they impact your response to food and brain. So there are sets of genes within the obese groups that actually can drive you to eat more fat or more sugar or to not feel full after meals. The understanding of the etiology of the specific components of obesity can help you at least understand how you'll be reacting in this environment, because the whole do not overeat statement is not working in public health at the moment. So I think the genetics and understanding the genetic etiology of your condition is very important. But as we spoke about earlier, actually the epigenetic component is important. It codifies or reinforces the signals. So epigenetics is not absent from this process. So if you think about, you've got increased eating of food, your genetics is driving you to overeat, you've got potential pathways that overexpress because of your genetics are then driving the increased eating of fat, for example, or sugar or whatever, then you can get epigenetic reinforcement of these pathways. So epigenetics can then fan the flames of this process. But also, again, going back to the conversation of mutations within these processes, there are genes that are mutated that increase the risk, or variants that increase the risk of obesity, that are associated with epigenetic regulation. And FTO is one of the most sort of famous ones. You've probably heard of FTO. So hopefully that was informative and can kind of show how epigenetics sits within the broad plethora of the biological processes that can drive you to have increased disease risk or obesity risk in this case. So yeah, epigenetic modifications change DNA expression. They're great for cell specification and do have a role in adapting the cell in responses to these environmental cues. However, they are not the primary driver. So when people say it's all about epigenetics, it's just not the true. The majority of the diseases that I think are these chronic diseases, and every day we see more and more genetic variants associated with diseases, are due to complex and very complex environmental genetic interactions. So really understanding how the environment impacts our behaviors and our metabolism, and the genetic lesions that then lead to those differential responses is key to understanding chronic disease. However, epigenetics is an important component of that, and understanding how it reinforces and as we say, fans the flames of these conditions is really important. Also, epigenetic signals and markers are very good methods. You can actually see the different modifications are very good ways of understanding what's happening in a cell as well. So epigenetics will become a very interesting biomarker, and you can see this within the aging field, where we've got these. I'll be brutally honest with you, the arrays are underpowered at the moment, and I think they have little utility. However, I believe that these epigenetic arrays in the future will be able to give us a lot of information, and you can see this with cancer biology. I mean, cancer is a disease where you get multiple genetic lesions, mutations that drive over proliferation, and then you get changes in the epigenetic landscape, which then reinforces and drives tumours to grow more viciously. So again, there's another great example, and the epigenetic signals within these tumour cells can inform people what's going on in these tumour cells and enable people to create better therapies because of the targets that are observed. So epigenetics is a very good process to codify what is being driven fundamentally by your environment and your genetics. It is not independent, and it does not sit above genetics, and it is not the primary driver of obesity. Thanks for listening.